enzyme in detail, where we have invested like 7 lectures on FOC1, and then after that we have invested 4 lectures, around 4 to 5 lectures on zinc finger domains, where we saw how it was purified, how it was known that this is transcribing 5S gene, or how it is important. Ultimately we saw the structure and how it is binding your DNA sequences. Now, since we are talking about binding DNA binding proteins, basically we are talking, and why it is important, I have mentioned it 2-3 times, that it is important because we want to create one artificial enzyme, or to say DNA binding protein, so that we can target our own target DNA. So we want to create a protein that can target specific DNA sequences, and slowly we are moving further towards it. See, we have seen some limitations are there in zinc finger proteins. Individually, it is very good, but some of the bases are not properly targeted because of some limitations in modularity. And also there are some off-target effects, because at first position or second position, there are multiple in nucleotides that can be recognized by the same protein. So because of that, same signal can be attached to either one site or to another site. And because of all this reason, one has to look further for the specificity of the enzyme. And so far, I have not talked about or even mentioned about meganucleases. So I would ask you to actually look for meganucleases, because these are also long nucleases that can bind specifically to longer base pair, specifically 8 to sometimes 18 base pair. And these are very useful, but it cannot be changed further because they are very, very specific in binding the targets. You cannot mutate this target sites. So that I will come to it later. But in today's talk, we are moving further to talon. So specifically, if we see in today's lecture, we are moving into the transcription activator like effectors. So this is named as TALE, or TALE. So this is for transcription activator like effectors, and I will come to it later. So the tail story starts with your gram-negative bacteria, and this is the plant system, right? So far what we have seen, either it is bacteria or xenopus, from where the transcription factor was coming. But in this case, it is coming from your gram-negative bacteria, and it actually infects different kinds of plant systems. So we will see how it infects and what is the story behind your tail. So basically, you have a gram-negative bacteria called Xanthomonas, which infect different kinds of plants. So this within this plants, you might have pepper plant, then you have rice plant, citrus, cotton, you name it, tomato, soybean. So there are various kinds of plants that can be infected by your Xanthomonas species. So that means Xanthomonas is a plant pathogen. Okay. So now there are different species of or strains of Xanthomonas, and these strains are grouped into different pathovars. And this pathovars distinction is based on the distinctive, pathogenicity of one or two more plant hosts. So these are the different pathovars of Xanthomonas campisteris. So these are classified, that which species, either it is infecting oryza, that means rice, or some citrus plant here, or another soybean plants, or some other plants. So based on that, or mustard. So based on this has been classified into different pathovars. Now, the infection, how it occurs, the infection from Xanthomonas into plants occur through natural openings. That means either stomata or hydatodes or natural wounds. So what you see here are the epidermis, lower epidermis, and what you see specifically in arrow are the microcolonies of your Xanthomonas bacterium, gram-negative bacterium. So here as well as here. So basically, you have stomata, which is responsible for carbon dioxide uptake and making of photosynthesis. But these opening are enough for your infection of these kind of bacteria, gram-negative bacteria. Now, after infection, actually they make these kind of spots. For example, what you see here are the black spots, or they are able to make streak marks, as you can see here. So basically, some bacteria gets access towards your vascular system, and then they spread through your plant's xylem and spread through systematically into the whole plant. Otherwise, they are restricted into leaf mesophyll system. So this is the thing. Now, what we are talking about here is the plant pathogen, and since it is infecting plant, so plant should also have some kind of immunity, right? So we will discuss little bit about plant and the immunity of plant and how these two are correlated. For example, plant has two major the kind of immunity. One is pattern triggered immunity and second is effector triggered immunity. So the pathogen what we are discussing today about is Xanthomonas species, and these are the virulence factor that actually forms your effector proteins. So we will basically see effector proteins coming from your Xanthomonas species and how plant generate effector triggered immunity. So this is the story between these two, effector triggered immunity in the plant. So how the field started? Actually, since the start, many people actually wondered that why there is certain kind of plant which are not infected by disease, but the same species are or look very diseased one. So actually, they wondered why certain plants have resistance towards some disease and why others are very susceptible, and what is the molecular basis behind this susceptibility or behind this resistance within the plant. So they in the quiz puzzled to many of the scientists until one scientist named Harlot Henry Floor. So this scientist was very methodical, and he was studying your cultivar named fox plant, and he actually studied this fog plant and the pathogen, that is fungus, which is infecting this plant, as in shown in yellow in this case. And he studied the genetic interaction. Basically, not only one plant, he took different species of fox plant and different variants of this fungus, and he was testing it. And finally, he actually proposed one hypothesis, that there is something called gene for gene hypothesis. So, gene for gene hypothesis for plant and pathogen genetic interactions. So according to him, what happens that if you have, so for him, what he figured out that the pathogen coming from the bacteria has certain kind of gene. He named it avirulence gene. AVR is for avirulence gene, and the plant which are resistant specifically towards this AVR gene carry some kind of resistance gene, is shown as a R. So this is the R gene. And basically, when you have both interactions happening, that means if you have AVR gene present and as well as you have resistance gene present from the plant, that means this two interaction happens and you will not get any susceptibility in this plant. That means this plant is resistance. Whereas in other cases, if you do not have any R gene is minus in this case, then this plant is susceptible. Similarly, if you do not have AVR but you have R but of different origin, that means this R is not cognate partner of AVR gene. That means these two are not, having any kind of interaction, then also this plant is susceptible. So this kind of hypothesis actually he proposed after working with your flux gene and fungus protein. And basically this was only hypothesis which came into normal experimental way only after 10 to 15 years from his proposition, there was a molecular proof for the existence of these interaction. So scientists actually started cloning different kind of genes. For example, the first AVR gene was cloned by Brian Watts here, together with Noel Keen lab, and they had cloned some airborne genes from Pseudomonas syringae. And slowly after that also, other people, many people started cloning our gene from the plant. So basically they were trying to see whether gene to gene hypothesis was true, and eventually they were able to find these two kind of protein. They were also able to clone AVR gene, and they were also able to clone our gene. So now, according to them, according to many scientists, they were thinking that from their prediction, that how these two AVR and your R gene must be working together. So from their proposition, original proposition, what they thought, that the pathogen, for example, here is Xanthomonas, is producing this AVR protein, and this is secreted outside the membrane of your xanthomonas. So that means this protein is secreted outside, and then these are cognately bound by your plant cell receptor system, which is present on the cell membrane of the plant cell wall, all the cell wall. So this was their proposition. So because of that, they have something like signaling pathway, and this kind of idea they got from your receptor signaling pathway. So if you have studied receptor signal pathway, you must be clear that this is something like that is very prominent in animal system, where you have certain kind of ligand and then you have receptor, and then followed that you get the different signaling map kinase pathway or other pathways are signaled, and then you get some certain kind of end product after going into the nucleus. So this was also proposed, and there was no proof for this kind of proposition. So, what they found, they started looking closely into the R gene as well as AVR gene. So, basically, when we will study here AVR, we will see that this was not true, and also the receptor signal, which they initially proposed that should be present at the membrane, which was also not true. So, slowly they started, getting some kind of R gene clone. So for example, here the N gene from tobacco against viruses infection, CF9 gene against fungal disease infection, or RPS2 gene against bacterial disease infection, and this MI gene against nematode root in nematode infection. They all had certain kind of repeats, for example, leucine rich repeats. And when they checked in the database that how these repeats are able to form, so they actually found some of the animal proteins, for example NOD1, which also has LLR region, and they compared that they all had LRR region. So basically what you are seeing here, that animal and plant have certain similar kind of proteins that are fending against your pathogen. So this was confirmed, and on top of it, it was known that certain middle region was acting as a nucleotide binding region. So this they actually found out from the database, and after that they came into the they came again back into the AVR gene. Now AVR gene, when they studied further, they did not find any signal peptides. So for example, if it had to be secreted from the xanthomonas, then they did not find any peptides which are, behaving as a signal peptide. That means it is not secreted. They did not find any secretory proteins here. So neither this is secretory, that means it is not secreted from the pathogen. And then when they found that receptor also is not bound here. So basically by this two examination, they were able to tell that AVR protein is not conventional, it do not have conventional secretion signal. So this should not be acting like a ligand receptor kind of molecule. So this proposition was not accepted. So next, what they found basically for our proteins, like mean the resistance genes. So they found that most of the resistance gene was actually placed in the cytoplasm. So not in the membrane region, but they were placed here into the cytoplasm. So basically, that is also giving one proof that they are not behaving like a ligand or a receptor kind of signals. Now, later it was found that AVR gene or the protein is prepared inside your cytoplasm, and then through the type 3 secretion system, it is also called translocon system. So from this type of syringe, these proteins are directly injected into the plant cell wall. So as you can see in the movement of your red proteins, it is first here in the bacteria and then slowly it is moving into the big plant cell cytoplasm. So it turns out that most of the AVR proteins are injected into the plant cell, and then they started finding out that what is the reason of sending these protein into the plant. So what you are seeing here in green are the resistance gene as shown here. So gene resistance gene, and what you are seeing here in red are the AVR effector proteins. So they actually match here. So if they are having cognate binding partner, then they will bind exactly in the sequences. If they are not having cognate binding partner, that means they will remain ideal and there is no binding towards its any cognate resistance gene. So basically this type 3 secretion system, if we see, they are highly conserved in humans as well as in plant pathogenic bacteria. For example, this is shown as a syringe kind of system, and when you see here, this is the syringe and the red acting molecule is your effector molecule, and that is getting inside your cytoplasm or cytosol. And this translocation of effector is also occurs in many other pathogens starting from fungi, oomycetes or nematodes. So basically the other systems also send their pathogens directly inside the cell wall. Now, what is the significance of this AVR proteins or AVR genes? First, I will tell you the partner of AVR and resistance gene. So there are two different kinds of way how AVR and R can be recognized. For example, first, the model plant in a plant system, if somebody is working in a plant, they already know that Arabidopsis is the model plant system. And when first Arabidopsis genome was sequenced, it was found that there are only about 100 R genes. So basically, there are only 100 R genes. But when it comes to pathogens, there are a lot of different pathogens which are, infecting or doing infection in the plant. So there are more than 1,000 microbes which might be infecting plants. So the question arises if there is a direct recognition, that means one AVR to one R gene, that means there are only limited amount of pathogenicity that can be fended by plant system or by R gene. So now how this limited number of R gene is able to recognize different AVR system. So it turns out that several AVR gene or protein is able to create different binding patterns. In this case, this RIN4 is one R gene. Okay, and this R gene via binding by AVRB and AVRPM creates another binding pattern. How? Because it is phosphorylating this RN4, and by phosphorylating this RN4, it is making a pattern like it is attracting RPM1 and protein. That means this is the uh, immune system, immune response kind of protein which is coming out. So that means by mutating RN4, not mutating, I should say, by creating another interaction pattern, it is allowing other molecules to be interacted with R gene. And by doing so, they are creating disease resistance. That means they are not anymore susceptible. So this way of recognition is called your indirect way of molecular recognition from AVR to R gene. And another example, you can get it from, this is another R gene, AVR gene. And in this case, when it binds to RIN4, it cleaves into two different parts. And by cleaving it, another molecule is binding to RIN4 here, and then it is creating your disease resistance. So there are ways in which your R gene can be recognized by different effector molecules, and hence they are generating a certain kind of immunity. So this was found like direct as well as indirect recognition is there. Now coming to the role of AVR genes. So basically if you see or if you think that AVR proteins are directly injected into your plant cytosol, then and it is recognized also by your plant system. That means it is also recognized by resistance gene. So ultimately what is the role? If it is recognized by our gene, resistance gene, then how it is advantageous to the bacteria by injecting this effector protein. So what is the major role of AVR gene? So that we'll see now. So it turns out that the major role for AVR is to suppress pattern triggered immunity. So basically, if you see here in the third one, this is the pattern triggered immunity. So you have some kind of recognition by plant system. Basically, it is recognizing your some molecules here from the flagella. So by recognizing this molecule, it is activating signaling pathway, for example mp map kkk pathway, and then it actually induces different kind of proteins which enhance the resistance in the plant. So that means you get a resistance system in the plant, and basically you are getting fending off any of the infection caused by the pathogen here. But pathogen is very smart. It has evolved in certain way that in this case it is not able to recognize its this flagella. In this case, let's take this, that this is flagella. So instead of giving its flagella as a recognition molecule, now it is behaving or evolved in such a way that it has evolved to produce a translocon system where it is directly injecting its material inside the cytosol. So once you have the material inside the cytosol, that means these effector molecules, they are not able to recognize by your signaling pathway. So that means these things are not able to recognize your effector molecules, and hence there is no immunity generated against this pathogen, and hence in the end your plant cell is susceptible to your infection. So this is how the advantage for AVR effectors. So that means if this plant is naive, that means it does not have any R gene. That means in that case, this can be injected into the material. That means effector molecules will be injected into the cytosol, and by injecting it, it will be suppressing your pattern PAMP triggered immunity response, and in that way it is killing your plant. So pathogen is able to kill your plants by making it more susceptible. But again, plant is again very smart, and it has evolved in other way. That means this plant has generated this R gene. For example, these are the R gene, and by having this R gene, you have these are gene which are again able to recognize this effector molecules. And once it is recognizing this effector molecules, then that means they are generating some kind of hypersensitive response system, and by doing so it is making its own plant system more resistant. So that means it is fending off all the pathogens which are infecting your plant cell. So this is how these two systems are generated, and basically the original function of AVR effector system is to kill plants via sending directly the effector molecules. But in this case, if the R gene is present, that means this R gene makes the plant more resistant toward the any infection. So that means based on these fighting, whether there are our genes or they are not our genes, or whether they are a cognate effector or our genes molecule. So based on this fight, one wins, either the pathogen wins or whether the plant cells wins. Okay, so this is the basic just we will see in the whole lecture. And what we have already seen, the stomata pore where your bacteria gets intracellular, and this is not the passive procedure. That means it is not just the passing through the stomata, but there is an active procedure, because all these molecules are recognized within your stomatal basically by guard cells, and they close these guard cells, and then you have molecular recognition, and immediately you have some kind of signaling molecules going on further, and that will recognize your cells. And why I'm saying so that this is not a passive, because it has been found through experiments that when you have these bacteria and you have a syringe, and without giving directly from the stomata, but if you inject it directly into the intracellular space within the leaf, then this doesn't create any hypersensitive response, but this only behaves as a wild type. So that means by infiltration. So this kind of method is called infiltration. Infiltration into the leaf, they do not generate any kind of response system. Okay, so so far what we have seen are the two branches of plant innate immune system that actually constantly working against your attacking pathogens, and based on these two, either pathogen wins or plant wins based on what kind of immune either pattern triggered immunity or effector immunity is induced. So this is the total overview what we are going to see in our lecture, especially for Xanthomonas and especially for talons. So how talons were generated, or how talons were actually seen for the first time. This is just a representation where Xanthomonas on the leaves are shown through electron micrograph pictures. And what you see here is again the translocon kind of system. So it is from the plasma membrane, inner membrane, outer membrane, cell wall, and then this is the inner membrane. And what you get directly the proteins here. So basically this Xanthomonas has a lot of different kind of effector molecules, basically more than 20 to 40 effector molecules. And out of these effector molecules, tail contain the largest family of effector molecules. That means transcription activator like. And why it is saying so, transcription activator, because through the picture you can understand that this comes into the cell wall, goes into the nucleus through some kind of piggy material, and then inside that it generates some kind of transcript which are important for further activity like immune response or other responses. Okay, so that is why it is called transcription activator like effector molecules. So once it was found, then in the group of Robert Stahl and team from the University of Florida, they were able to generate some resistant plant. So for example, ECW30R is resistant against one, pathogen, that is AVR, avirulent gene BS3. So this is the gene BS3, AVR, avirulent gene BS3. And the same he created a resistant variant by incorporating a dominant R gene, resistant gene incorporating BS3. So this is the example of gene to gene hypothesis. Whereas when he incorporated BS1, which is not the cognate partner of BS3, avirulence BS3, then this plant was susceptible. So by creating this kind of system, he was able to show that you have avirulent gene BS3, which is making ECW30R resistant, whereas other plant having ECW10R, that is carrying resistant gene of non-cognate partner, is susceptible and is not resistant towards the disease. So um, how this was found? So initially what happened, so you can imagine this is the bacteria, and you have certain nucleus, and these are the genes. So basically these are found in 20 HRP gene. And when you see this system, I've shown it already here. So from inner membrane, outer membrane, and then cell wall, and as well as plasma membrane, it goes into the cytosol. So this is the protein peptide, it has been shown. And after going into the cell, cytoplasm, it has two different kind of fate. So either it is not recognized by our gene because this AVR is not cognate to this R gene, and AVR3 is very much when it finds its cognate R gene, then it creates a hypersensitive response. And what this response is physically in sense, how you can see that is shown in this picture. For example, so in first case, so this is the example where the effector gene is coming into the cytosol, but it is not meeting any of the R gene. So for example, there is no cognate R gene. In that case, your plant gets diseased and it has the symptoms like water soaked lesions. So what you see here are the some kind of water sogginess, and then it becomes black and other things. So it gets molted afterwards. So this is the system when it is not resistant. Whereas once it finds the resistance protein inside the plant and it is getting recognized by the R gene inside the plant, then you get a hypersensitive response. And this hypersensitive response means you have a local death of the plant, specifically the place where you are getting infection. So this is the local plant death, and by doing so, it actually halts bacterial progression through this system. So basically, if this region is died, and this region are not there, so that means there is no progression of the bacteria. So they were able to clone this AVR gene, and initially after cloning they found the first observation was the gene was very big compared to bacterial genes. This gene was around 5 kb, 4.3 to 5 kb, and they said that this is very 5, and they were able to clone within the segment. And initial sequencing they wanted to do, so initial sequencing was unsuccessful because there was lot of GC content in that. More than 94 to 96, sorry, 64 to 67% of genes were having GC rich content. So what happened? They then cleaved this portion into bits and parts. For example, if you see they made new mutants or they made smaller mutants also. And then they started giving it for maximum Gilbert kind of sequencing. And from maximum Gilbert sequencing, they were able to see that whatever sequences were coming out, they looked exactly similar, whether the smaller pieces, longer pieces, or especially the middle region of especially the middle region of the whole segment repeats again. So they did the sequencing, and initially in the first paper, so what this shows here is the ORF region. So basically they wanted to check also after cloning that which of the side is important for promoter, whether the gene is prepared from this direction as in case of ORF, or this is prepared from left direction as it's shown in this ORF2 or ORF3. So once they started, sequencing this, they were surprised to find that most of the middle region showed similar kind of sequences. For example, I have also colored it in different colors. So for example, here LTPEQVV, LTPQ, this is the first few amino acid I'm showing, but the underlying region here LTPQEVV until this LGHG. So this was repeated several times within AVR gene. So, and they just reported in their first paper, they just reported these sequences, and in the discussion they wrote that whether this makes any sense or whether what is the significance of these repeated sequences has to be found in the biological evolution. So that was their first observation, that they have found AVR genes and how they are significant. Next, they were able to, uh, align this, and what they from this data, another paper, they were able to show that this gene was repetitive and the repetition contains 102 base pair, and within this 102 base pair there are 34 amino acid which is already always there in the repeat sequences. So basically you have a repeat of 102 base pair, and these are repeated several times. For example, in this case it is repeated 17.5 times, because in 18 repeats you will see the repeats are only conserved until this phase, and the last 5 phase is not conserved, only in few paces. So it is conserved from 17.5. So then they processed further and they performed some expression kind of studies. So especially in plant you have some GUS kind of reporter. So GUS is a beta glucuronidase gene, and it is mostly used in the reporter system kind of assay. So basically to find out promoters or to find out expression kind of studies. So what they used here, so they used sequences nearby your AVR gene, BS3 I will write, and this is from 1 to 700 base pair. They used this and reported with the GUS gene. So here with the help of this they were able to find all this data, means they are able to express it. So this is expressing. Then again they deleted 500 base pair from the side and this was able to express. But if they delete further down, further till here, that means promoter activity lies until your 503 base pair from here to here. That means until 200 base pair, your promoter activity is still intact. But if you delete further, the promoter activity goes down or not there, and you do not see any expression, no expression in this case. Similarly, they wanted to check whether there are any promoter region in this side from the left side or the non-coding end. So they also cloned this region and they were able to see that there was no expression. So basically all the expression was coming from the right hand side and then the top one. So then that means you have the promoter activity known. And also they cloned this, expressed this protein, and from the expression they were able to find that these expressions are based on 122 kilodalton kind of protein. So this is the delta construct of one of the repeats. So they were able to find different constructs here and they were able to express this protein in SDS gels. So next, what was the reason? Since they were finding this from AVR3, now they actually checked other AVR genes. For example, in this case, from different sources, they were able to find homologs, AVR XA5, AVR XA7, and AVR XA10. So basically, they uh, prepared conserved sequences from AVRBS3 that was originally found, and then they also found some of the sequences from your rice species, and they found all this pathogen, they sequenced it, and what they found that again you have 102 base pair repeats, that means you have 34 amino acid, and again these repeats are there. But based on your homology, it was found that the lot of conserved regions are there, and this is the hypervariable region they found. So this is the main region, and this region always occurred at 12 to 13th positions. So we will see next slide. So after this finding, maybe I conclude here. We will start from tomorrow's lecture. This is not complete. Today's lecture is not complete. I will start from this slide number 40 and we will go further. Is that okay?
Yes, ma'am.
Because I feel I'm going very fast, and we can do it tomorrow in next lecture. Okay, so we will continue this further. That the gene is cloned, and we have seen this cloning, and I will just do one slide. So there was a lot of things. So first we have seen how it is secreted. There was no signals. Then the tail is the largest transcription activator, and then gene to gene relationship we have seen so much, and we will see tomorrow what next lecture how the homologs were generated, and then mutational studies on homologs repeat order. Okay, All right. Thank you so much. See you in next class. That is on Tuesday, I guess. Okay.
Ma'am, just a
Okay.
confirmation.